All right, today we're going to dive into what's basically a genetic detective story. It's this awesome tale about how a broken rule and a really puzzling mystery led to, well, the very first maps of our own DNA.
And this idea right here, it's the key to the whole thing. You know, in science, it turns out the most exciting stuff, the real game changers, they don't happen when you prove a rule is right. They happen when you find an exception and have to figure out why it's so spectacularly wrong.
So, let's set the scene. Our story kicks off with what was, for a long time, the law of the land in genetics. I'm talking, of course, about the foundational work of Gregor Mendel.
Okay, so Mendel's second law, the law of independent assortment, sounds complicated, but it's actually beautifully simple. It just says that when you're looking at different traits, say a plant's height and its flower color, the genes for those traits get sorted into egg or sperm cells completely independently of each other. The old analogy is kind of like sorting laundry, right? Your socks go in one pile, your shirts go in another. They don't influence each other.
And here's the cool part. This law came with a built-in prediction. A signature, really. If you did a specific kind of cross, a dihybrid cross for you genetics buffs, you were supposed to get this super clean, predictable 9:3 to 3:1 ratio of traits in the offspring. Every time. It was the gold standard. I mean, it just worked. Until it didn't.
And that brings us to the crime scene. This is the moment where a couple of scientists stumbled onto something that just didn't make any sense. The results didn't add up, and it created one of the first great mysteries in genetics.
So back in the early 1900s, these two researchers, Bateson and Punnett, were doing their own experiments, and they found something totally baffling. Some pairs of traits just refused to follow that neat 9:3 to 3:1 rule. It was like their genetic laundry wasn't sorting right at all. Some socks were always ending up in the shirt pile. The rule was clearly broken.
So the big question became, if genes weren't always assorting independently, then what in the world was going on? I mean, if Mendel's elegant law could just be broken, it meant our whole understanding of heredity had a massive hole in it. The investigation was officially on.
And this is where our detectives enter the story. A team over at Columbia University came up with a pretty radical new idea. An idea that wouldn't just solve this mystery, but would blow open a whole new field of science.
First up was T.H. Morgan. He had this breakthrough thought. What if these genes aren't independent because, well, they're not independent? What if they're physically stuck together or linked on the very same chromosome? And then, get this, one of his undergraduate students, a guy named Alfred Sturtevant, had an even bigger flash of genius. He realized that how often that link gets broken could actually be a way to measure the physical distance between the genes. So let's break down what that actually means. If genes are unlinked on totally different chromosomes, you get 4 types of egg or sperm cells, and they show up in pretty much equal numbers. Simple enough. But if the genes are linked on the same chromosome, they tend to travel together like buddies. This means you get a ton of offspring that look just like the parents. We call those parental types. And you only get a few with the new, shuffled-up combinations, which we call recombinant. But that just leads to another question, right? How do linked genes ever get separated at all?
Well, the answer is this incredible, intricate biological dance. It's an actual physical process that literally shuffles the genetic deck. It's called crossing over. And you got to understand, this isn't just an abstract idea. It is a real physical thing that happens inside cells, where chromosomes, the ones you get from each parent, line up and literally swap pieces. This is the how-done-it of our whole mystery.
It happens in 4 basic steps. First, the chromosome pairs get real cozy. Then, their arms physically cross over each other at a spot called a chiasma. And right at that point, they break and exchange entire segments of DNA. What's the result? You get brand new chromatids that are a mix-and-match combination of alleles from both parents. This is how genes that start out on the same chromosome can get shuffled around.
Okay, so here is the absolute crucial takeaway. Imagine you have 2 dots on a piece of string. If those dots are super close together, it's pretty hard to find a random spot to cut the string between them, right? But if they're far apart, it's easy. It's the exact same idea with genes on a chromosome. The closer 2 genes are, the less likely it is that a random crossover event is going to land in that tiny space between them. And that realization, that was the key that unlocked everything.
Because think about it. If the distance between genes changes how often they get separated, then you can flip that idea on its head. You can use how often they get separated to figure out the distance.
This was Sturtevant's brilliant, incredible leap of logic. He realized you could just count The number of recombinant offspring, the rare ones with the unexpected trait combos, and that number would tell you how far apart the genes are. The percentage of those recombinant offspring could be directly translated into a distance on a genetic map.
And out of this idea came this really simple but incredibly powerful formula. You just take the number of recombinants, divide by the total number of offspring, and multiply by 100. That gives you a number in map units, which is your genetic distance. So let's grab our detective's toolkit and see how it works on a real case.
All right, check out this data from a rabbit cross. We're looking at 2 genes here, one for spots and one for hair length. And right away you can see that 2 of these groups are way more common than the others, the 75 and the 66. Those are our parental types. That's the original combination the parents had. And then you have these other 2 groups, with only 10 and 7, they're much more rare. These are our recombinants. They're the evidence that a crossover event happened.
Okay, time to do the math. Let's plug in the numbers. We add up our recombinants. So 10 plus 7 gives us 17. We divide that by the total number of offspring, which is 158. Multiply that by 100, and bam! We get our answer, 10.8. What does that mean? It means the gene for spots and the gene for hair length are 10.8 map units apart on that rabbit chromosome. The case is solved.
So, why do we care so much about this one little puzzle with rabbits and pea plants? Because solving it was a huge deal. It gave scientists the very first tool they ever had to make genetic maps, to actually visualize the invisible layout of a chromosome. This fundamental technique, born from a broken rule, paved the way for decades of research. And yeah, it eventually led straight to the massive, world-changing Human Genome Project.
So think about that for a second. A simple observation that a rule wasn't working right led directly to our ability to map our own genes. It really makes you wonder, doesn't it? What other massive secrets are hiding out there, just waiting for someone to notice an exception to the rule?